Good afternoon to all the EAPCET aspirants. So here we are in our second session of the chapter Principles of Inheritance, which is the most demanded chapter by many students. So here we are in the concept of codominance, wherein both the alleles are equally dominant, are equally expressing their phenotypic expression. So here, once let us recall the concept of in this then the concept of codominance will be clear. So first, if you observe, yesterday we have learned Mendel's law of dominance and law of segregation. In law of dominance, we have clearly noted that each factor occurs in pairs with contrasting traits with one dominant and one recessive allele. Dominant allele will always inhibit or mask the effect of recessive allele. That is the concept of Mendelian inheritance. Law of dominance. Okay, so in incomplete dominance, what happens is the dominant allele is not completely masking the effect of recessive allele. That is the reason the dominance phenotypic expression range will be decreased. That we have observed in the flower color of snapdragon or antirrhinum, and we have also observed that in Mirabilis jalapa, which is 4 o'clock plant. And in codominance, how is this concept deviating from Mendel's law of dominance? This is deviating as such, where both the alleles will be equally dominant. Okay, both the alleles are equally dominant. Fine, right. Best example we can take is ABO blood grouping. ABO blood grouping is an expression which is coded by a single gene, one gene, that is I gene. Okay, so this I gene, whichever is present in the RBCs, RBC cell wall, let us suppose this is RBC, yeah, whose wall layer will have different polysaccharide units. If at all it is IA, this one gene, I gene, can have 3 alleles. They in the form of IA, IB, any antigen. So based on the polysaccharide which is present on the RBC surface, here antigen will be decided. If at all the surface of RBC is having NAG unit, that is N-acetyl glucosamine. N-acetyl glucosamine. That is IA grouping, which is of A antigen. A antigen. Fine, right? And if at all the surface of the RBC is IB, it will be having the polysaccharide unit, which is galactose. Galactose. Fine, right? Which is beta galactose. This polysaccharide structure or sugar unit which is present over the RBC surface will make it IB. It will make it B antigen. As such, IA and IB are dominant over IG, whereas IO blood grouping will not have any antigen over its surface. So here if you observe, whenever IA and IB blood groups are present together in AB form of blood grouping, both IA and IB genes are present, both the alleles are equally showing its expression. So then AB blood group has come into picture. So here if you observe it clearly, in ABO blood grouping, here you have NAG units, which is N-acetyl glucosamine, making it IA antigen. If the RBC surface is having galactose, it is IB blood group, okay, which is having B antigen. Both of them together will be present in AB blood grouping, showing their expression equally. Here comes the concept of codominance, where both the alleles are equal in their expression. So what type of questions can we expect from ABO blood grouping? Look at here carefully. For example, the allele from parent 1, let us suppose, is having IA allele. Okay, allele from parent 1 is having IA allele, allele from parent 2 IA itself. Then IA combination, it will give you A blood group. Okay, it is A blood group. If IA and IB are present, both the alleles will show the expression in the offspring, giving rise to AB blood grouping. Okay, this will give rise to AB blood grouping. Only IA and IO, where IA is dominant over the IO, it will mask the effect of I gene, giving out just A blood group. Okay, and it will have A antigen itself. Same the case goes with AB blood grouping and B blood group also. IB will give rise to B blood group. IB, IO, wherein IB will be dominating over IO, it will give rise to B blood group. And no antigens above it, let us suppose I gene or I allele from both the parents will give rise to O blood group. IO will give rise to O blood group. The combinations of the alleles from parent and what genotype is expected in the offspring resulting in what blood group is important. So, this is the concept of ABO blood grouping, which is ruled by single gene. This single gene is having multiple alleles. So, this will also stand as an example for multiple allelism. Multiple allelism is also deviating from Mendel's law of dominance. How? Because according to his concept, one gene is coded by two alleles, but here in multiple allelism, one gene is having many alleles. As such, multiple allelism is also deviating from Mendel's law of dominance, and here ABO blood grouping is also considered as an example for multiple allelism with respect to the gene having three alleles. And this is also an example for codominance with respect to ABO blood group. Clear till here, ma? You are clear with codominance and multiple allelism concept with examples, right? And make sure you are clear with the allelic combinations of both the parents with resulting genotype blood group in the offspring. Now let us clearly understand the differences between codominance and incomplete dominance. Codominance, you look at here carefully. Two parents' phenotypes are expressed together in the heterozygous offspring. Here in incomplete dominance, two parents' phenotypes, only one allele cannot mask the effect of the other allele. There is no mix or blend of two alleles, but here one allele cannot mask the effect of other. Clear, right? Here in codominance, two alleles are neither dominant nor recessive. Here one allele is incomplete dominant in incomplete dominance. The name itself specifies the phenomena here. Here there is no blending or mixing of the parent traits. Here also there is no blending or mixing. Hybrid results in the formation of a new phenotype in codominance. Here also it results in the formation of a new phenotypic expression. And here phenotypic ratio and genotypic ratio is 1:2:1. Here also phenotypic and genotypic ratio is 1:2:1. Clear till here? Any doubts anywhere, ma? Yeah. Multiple allelism is also clear to us. Now we need to learn one more concept which is deviating from Mendel's law of dominance, that is pleiotropy. One gene is showing one phenotypic expression according to law of dominance. One gene will show one phenotypic expression. But here according to pleiotropy, in this concept, you will clearly observe one gene shows multiple effects. Multiple phenotypic expressions or multiple effects. Best example I can give you is skin depigmentation of Drosophila.
No biopsy is given now, and this do not take action. Here is normality into V. So both should be either 8 or both should be ml. Okay. So
Fine.
and into N factor. So what is
Skin depigmentation of Drosophila followed by starch synthesis.
medium means? Converting into plus 2. In basically, in neutral medium it is, in basic medium it is,
Phenylketonuria
this
disease
is what you need to remember.
is autosomal recessive.
Equals, say, ion 15 ml 2 into 10 power minus 2 molar.
Ketonuria, how does this disease occur?
Okay, so now what?
The aromatic amino acid, that is phenylalanine, has to get converted to tyrosine to show its expression. Okay, it has to convert to tyrosine by the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase. But absence of this one enzyme, when this one enzyme is inhibited or this one enzyme is not showing its expression, phenylalanine will get stored in the form of phenylpyruvic acid. Phenylpyruvic acid. This
Place of number of EQ1, we can also
of phenyl
into
many phenotypic expressions like mental retardation, frequent micturition, skin depigmentation, okay, and kidney sort of diseases and nervous issues. All these effects are caused just because of the absence of a single enzyme. One enzyme, one gene concept says that one enzyme is coded by single gene. So when a single gene is affected, it is showing multiple effects like mental retardation, frequent micturition, skin depigmentation. When all these effects are showed, it proves that the effect one gene is showing multiple effects, multiple phenotypic expressions. So here is the concept of phenylketonuria, which is explaining pleiotropy. Remember all of them carefully, okay. So what are the deviations? What happened in the story here? Whatever we have learnt in the first half. From monohybrid cross, Mendel has derived two laws. One is law of dominance and second is law of segregation. Law of segregation is universally accepted. Law of dominance is not universally accepted because there are many intra-allelic interactions, that is intragenetic interactions, which are deviating from laws. Those include pleiotropy, multiple allelism, codominance and incomplete dominance. All these concepts with examples we have learnt. Now, let us look into the concept of dihybrid cross and what are the results of the dihybrid cross, what is the law that is derived from dihybrid cross. In dihybrid cross, if you observe, in parental generation, two traits were taken, two characters are considered here. One is seed color and then seed shape. Here what are considered? Two characters.
Number of moles into its gives
And
in the place of number of into wing meters or
from parental generation, of course, true breeding lines are taken where capital R capital Y and small r small y are considered, and here how many gametes are formed? Two gametes, okay, where capital R Y is one gamete, small y is one gamete. These are the gametes, and in F1 generation, if you see, same the case that you have observed in monohybrid cross, same thing you will observe here, that all the offsprings of F1 generation are dominate phenotype only. Dominant phenotypic expression here is round seed and yellow seed color, okay, round and yellow seed color. With respect to seed color, dominant and recessive traits. With respect to seed shape, dominant and recessive. Dominant seed color is yellow. Recessive seed color is green. Dominant seed shape is round. Recessive seed shape is wrinkled. Okay, so here round and yellow are being taken. And in F2 generation, if you consider F2 generation, when F1 generation is selfing of F1 generation is of this genotype. How many gametes are involved here? How to calculate types of gametes? Remember students, to calculate types of gametes, the formula is 2 to the power of n. 2 to the power of n is the formula to calculate the types of gametes, where n is number of heterozygous alleles. Here how many heterozygous alleles are there? 2. 2 heterozygous alleles, so 2 to the power of 2 will be 4 gametes. So each parent is providing 4 gametes here. What are those 4 gametes combination? Look at here, capital R, capital Y, small r, small y, capital R, small y, small r, capital Y. These are 4 different combination of gametes. So here you have got the number of gametes, and gamete square will give you number of zygotes formed. Here 4 gametes each you have got, right? Now gamete square will give you the number of zygotes. Fine, right? Number of zygotes formed. Gamete square will give you the number of zygotes formed. Gamete square, if you consider 4 gametes each, right? So 4 into 4, on total 16 zygotes are formed, or 16 offsprings are formed in F2 generation, which are presented in a Punnett square. So that we will observe here. Just look at here carefully. I hope the complete explanation is clear to all of you. Here you see for dihybrid cross, 2 characters are considered, and the contrasting traits for each character, if you see, dominant seed color yellow and green is recessive, seed shape dominant is round, recessive is wrinkled. And here in the gametes you consider 2 gametes, and in F1 generation you will get heterozygous genotype and dominant phenotype, right? So in F2 generation, if you see, how many gametes are involved in the cross of self-pollination? To identify the number of gametes involved or types of gametes involved, the formula is 2 to the power of n, where n is the number of heterozygous allele. Let us calculate for each parent. Here heterozygous alleles are 2, so 2 to the power of 2 is 4 gametes each parent is, from each parent 4 gametes are involved. Now how many number of zygotes are formed in F2 generation? For that, to identify number of zygotes, the formula is gamete square. Gamete square 4 into 4 will give you the number of zygotes, that is 16. Now what are the possible combinations of these 16 zygotes that you have got? You have to put it in the form of phenotypic ratio. In that case, you can clearly observe from the Punnett square, 9 will be round and yellow, 3 will be round and green, 3 will be wrinkled and yellow, 1 will be wrinkled and green. Of 9, if you consider round and yellow, what is the genotypic ratio? Genotypic ratio will be very long, but then here for your calculation part of questions, you have to be clear with this structure of this 9 round
minus 3 divided by gone. Okay, so where it is gone, you have taken liters and ignored 1000. That's why it is wrong. Okay, now is it clear what we are doing? Yeah. So now tell me, this one 5 and 8 is there. So
you will get
1, so 3
of round and yellow seed. And next round and green. How many combinations here? 1 will be homozygous round and green, and 2 will be heterozygous round and homozygous green. Of course, the recessive trait will be in the homozygous form only. Next, 1 will be in the homozygous and homozygous green and yellow, and 2 will be homozygous wrinkled and yellow. 2 will be homozygous for the recessive trait and heterozygous for dominant. And rest 1 recessive for two traits will be small r, small y, small y. Fine, right? These are all the possible combinations. You have to remember it for sure. That is very easy to remember. You can easily draw it out beside your Punnett square also in your NCERT, which will be easy for your revision. Okay, and these combinations will be asked in your exams quite a times in your NEET PYQs. Such questions have been asked. Okay, so you have to be clear, from 16 zygotes or 16 offsprings, if 4 are heterozygous dominants, let us suppose in your question 120 seeds are asked. From 120 seeds, how many are heterozygous dominant? You can easily crisscross and find out the answer. So these ratios should be clear in your mind. Draw it out beside your Punnett square. Thank you, ma. Please continue with the class. Thank you, ma. Please continue with the class. If you are having any doubts anywhere, please drop it in the comment section. I'll answer it. Okay. And if you want any concepts in specific also, you can ask me. I'll answer it. Okay. I'll explain it. So this is all about dihybrid cross. Now, law of independent assortment. From law of independent assortment, this is the law which is dihybrid cross. From monohybrid cross, two laws were driven out. One is law of dominance and second is law of segregation. From law of independent assortment, the law which is driven out is law of from dihybrid cross, the law which is driven out is law of independent assortment. What is this law stating out? From two combinations, from when two characters are crossed, the inheritance of alleles is independent of the other combination of alleles. Okay, the inheritance or the transmit of one pair of alleles is independent of the other pair, is what given out in law of independent assortment. Gametes carry all the possible combinations of factors in equal frequency. When two different traits are involved, 4 gametes are formed. How did we calculate that 4 gametes are formed from 2 heterozygous alleles? It is from the formula of 2 to the power of n. When 2 heterozygous alleles are involved, 2 to the power of 2 will give you 4 gametes. So the transmission of one pair of alleles is independent of the other pair of alleles. That is what given out in law of independent assortment. Clear with this, right? So you have to be clear with all the possible combinations of gametes from this respective two different combinations. Okay. So here if you see law of dominance, law of segregation, which is also known as law of purity of gametes, and third is law of independent. Here law of dominance, this law states that in a cross between two organisms pure for any pair or pair of contrasting traits, the character that appears in F1 generation is called as dominant, and which is suppressed is known as in other terms. Segregation, when two different combinations are present, they are not present, they are not blend or mixed in that particular combination. For example, two heterozygous alleles are there existing together. Both of them are not blending or mixing with each other. That is law of segregation. When contrasting traits are crossed, the movement or transmission of one pair of alleles is independent of the other pair, is said in law of independent assortment. I hope all the three laws are clear to you. If you are having any doubt, you can ask me. Done ma, we will do that. In the playlist, if you can see, in the EAPCET playlist, all the chapters are organized in sequence only. Once you check out in the playlist, you will get all the classes in sequence. Okay, in the YouTube itself, if you check in live classes, you get different classes of all the subjects from our channel.
One more question.
Playlist, each subject wise playlist will be available where you will get all the chapters of each subject in detail. Okay ma? Yeah. Now polygenic inheritance, like how we have intra-allelic interactions, here we are learning about non-allelic interactions, which is deviating from Mendel's law of independent assortment. One such non-allelic interaction we are learning is non-allelic interaction. Is polygenic inheritance. Here, according to polygenic inheritance, traits are controlled by two or more genes. If you see pleiotropy, one gene is showing multiple effects. If you see polygenic interactions or polygenic inheritance, many genes are controlling one character or one trait. The trait controlled by two or more genes, that pattern of traits are known as polygenic traits, and when they are inherited, inheritance of polygenic traits are known as polygenic inheritance. As one gene is coding for many, one trait is controlled by two or more genes are involved in the inheritance. This is also known as quantitative inheritance, and the genes involved are polygenes, of which dominant alleles in the genes will show combination effect, the combined effect, whole effect is shown by the dominant alleles. Such effect is known as additive effect or cumulative effect. For example, what are the traits that are passed out as polygenic inheritance? For example, skin color, if you take as one character, you can see many variants of skin color, from negro to very lightly faired people. You can see many variants of skin color. This is controlled by polygenic inheritance, where this single trait is coded by multiple genes. That is the reason you will get a new phenotypic expression other than the parental expression. As such, you will have many variants. Next is height in humans, also is having many variants. Many variations of heights are seen. Kernel color in wheat. In wheat, if you see the kernel color, whole lot of variants of kernel colors are observed in wheat grains. Okay, so this is about polygenic inheritance. Now let us take the parental generation. Is the voice clear now, ma? Is my voice clear now? Just a second. Is my voice clear now? So here you see in the parental generation, only pure lines are being considered, recessive and dominant. Remember, small a, small b and small c, all these code for recessive traits or recessive alleles, and this is dominant. Okay, and the example we are taking here to understand is skin color. Fine. So here in F1 generation, of course, we will get the heterozygous alleles. Self-fertilization of this recessive, this intermediate alleles of F1 generation will give you, like, different combinations are possible when two individuals of same genotype are crossed, of intermediate genotype are crossed. There are so many combinations which can be expected. If you can see, first is complete dominant alleles. Capital A, capital B, capital C, capital B, capital C, capital C. All the dominant alleles will give you negro or black color. And whenever by the reduction of one dominant allele on the each end, here one recessive allele is added, here two recessive alleles are added, here three recessive alleles are added, here four are added, only two dominant alleles you can see, here only one, here completely albino. So when the reduction of one dominant allele is seen, complete dominant alleles are showing the additive effect of dark color, negro or black. Okay, with the reduction of one allele, one dominant allele in each step, we can see very dark, mulattoes, fair, very fair and albino. As such, the pigmentation is decreased with the decrease in the dominant allele number. Okay, here you can see 5 dominant alleles are there, wherein very dark color is observed. Here 4 dominant alleles dark, 3 dominant alleles mulattoes, only 2 dominant alleles fair, 1 dominant allele very fair, no dominant allele at all, albino. Fine, right? These are different allelic combinations. And here complete only dominant alleles are showing the additive effect. Extreme phenotypes, be it completely negro and black and completely albino. Extreme phenotypes are always rare, whereas intermediate phenotypes are more frequent. Understood, right? Next. So if you can take a graph of skin pigmentation with the F2 generation frequency, you will get a bell-shaped graph with the phenotypic ratio of 1:6:15:20:15:1. Completely a bell-shaped graph you will get, or a bell-shaped curve. Remember, the type of curve is also important. Now, to calculate the number of phenotypes in polygenic inheritance, the formula used is 2n plus 1, where n is equal to number of polygenes. Number of genotypes, to calculate the formula is 3 to the power of n, where n will be equal to number of polygenes again. Clear with this? And then Mendel's 3 laws of inheritance, we have monohybrid cross and dihybrid cross. Monohybrid and dihybrid cross. Here law of dominance and law of segregation are the laws which are derived from monohybrid cross. Law of independent assortment is the law which is derived from dihybrid cross. Deviations from monohybrid cross you can see incomplete dominance, codominance, multiple allelism and pleiotropy. Deviations from law of independent assortment, wherein multiple genes are dealing with one phenotypic expression, that is learnt under polygenic inheritance. Clear till here, ma? Now, see, these are the ratios. You can clearly see type of cross, phenotypic ratio and genotypic ratio. Monohybrid cross phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. Dihybrid 9:3:3:1 is the phenotypic ratio. If you square 1:2:1, that will give you the genotypic ratio of dihybrid cross. Test cross, it is always 1:1. Incomplete dominance and codominance, both genotypic ratio and phenotypic ratio will be 1:2:1 only. And polygenic inheritance we have just now learned, which will show a bell-shaped graph of 1:16, 15, 20:15, 6:1. This will be repeated with the reduction in the dominant allele number. Okay, so clear, right? Any doubts anywhere? If you want, you can take a screenshot of this, which is very important. Now, these are the most important formulae to be noted. Okay, one is types of gametes. To identify number of zygotes, it is gamete square. To identify number of phenotypes, it is 2 to the power of n. Number of genotypes, it is 3 to the power of n. For multiple allelism, to calculate number of phenotypes, it is n plus 1. Number of genotypes, n by 2 into n plus 1. For polygenic inheritance, we have already learnt it. Okay. Here completely n will be equal to number of heterozygous alleles. Okay. So these are different formulae to be noted clearly. Okay. Take a screenshot of it and apply in your problems. Sure, ma, I'll share you in the description. Right away I'll share you. The link is here, ma, you can go through it. Fine. So now, what are the key takeaways of today's session? Yeah, the key takeaways of today's session include codominance, incomplete dominance, multiple allelism, pleiotropy, followed by a brief account on law of dominance, segregation, law of independent assortment, by clearly noting out dihybrid cross with all the phenotypic expressions, all the genetic combinations, genotypic combinations from the phenotypic expression, and clear tabulated version of all the phenotypic genotypic ratios and clear tabulated form of all the formulae that we are using in it. And then we have also looked into polygenic inheritance by taking the example of skin color. These are the key takeaways of today's session. In the next session, we are going to learn on the why Mendel's laws have been deviated and what is the problem of the publications, followed by how chromosomal theory of inheritance is given out by Sutton and Bovary, the proof, experimental proof for chromosomal theory of inheritance, followed by mutations, genetic disorders, pedigree analysis, sex determination. These are the concepts that we are left over with, that we will do it in the next class. Till here, everything is clear, right, ma? So yes, that's all for today's session. Thank you.